Mastering Biochemistry for CSIR NET Life Science 2024 Essential Concepts and Tricks. Welcome viewers of Biotecnika. Today's video is going to be helpful to all the aspirants who are preparing for life science competitive PhD exams like GATE, DBT, ICMR, ICAR, CIFR and JAM. Let's look into what are the most important concepts for this particular subject and how you can prepare them well for scoring more in the exam. Today I have come with some important topics like stability in biomolecules, nucleic acids, metabolism, thermodynamics, enzymology, amino acids and protein structure. We will be starting with the first point, which is stability in biomolecules. What kind of biomolecules do I mean? I mean amino acids, proteins, nucleic acids, carbohydrates, vitamins, lipids, etc. All these macromolecules, they are coming from monomers which polymerize together to form bigger molecules in which there will be some stabilizing interactions. What you need to know from exam point of view is the different types of these intermolecular interactions. One such type is electrostatic interaction, hydrogen bonding, hydrophobic interaction, and Van der Waals interaction. What is electrostatic interaction? It is the attractive force between oppositely charged molecules or atoms. Hydrogen bond is a special type of dipole-dipole interaction, which involves a hydrogen atom bonded to an electronegative atom, which is example oxygen or nitrogen, and a nearby electronegative atom. The result is a partial positively charged hydrogen which is attracted to the negative charge of the electronegative atom. This provides significant stability to the biomolecules, especially the DNA and the proteins. The next type of bonding is hydrophobic interaction. This is not really a bonding, it is a type of interaction between non-polar molecules. They will cluster together in an aqueous environment to minimize their contact with water molecules. How is this achieved? It is driven by entropy. As the ordered water molecules surrounding nonpolar molecules, they become disordered. This arises due to the tendency of nonpolar molecules to avoid contact with water. Important in maintaining the structure and stability of biomolecule. For example, even the membrane formation where the phospholipid bilayer is formed by hiding the hydrophobic fatty acid tails inside the membrane. The last one is Van der Waals forces. We have different types. For example, between two polar molecules, also called as Kizom forces. Example will be between two water molecules or between two HCl, any two polar molecules. Ion dipole. It is the interaction between an ion and another polar molecule like sodium chloride in water. Dipole-induced dipole, also known as Debye forces, which are present between a polar and a nonpolar molecule. The dipole induces a temporary polarity in the nonpolar molecule, like HCl and Cl2, where Cl2 is considered nonpolar, but HCl temporarily induces a polarity in this molecule, thus both of them will stay together. Ion-induced dipole. It is the interaction between an ion and a polar molecule. The ion induces polarity in nonpolar molecule. Whereas here it was a polar molecule inducing polarity. Here it can be any kind of ion which induces a temporal polarity to the molecule which is nonpolar. For example, in oxygen. London dispersion forces, this is between two non-polar molecules, out of which one will be instantaneously created dipole, another one will be induced dipole. For example, in any two non-polar molecules like fluorine molecules. Coming back to the next essential concept, that is nucleic acids. Nucleic acids are commonly two types, mainly DNA and RNA, deoxyribonucleic acid and ribonucleic acid. It can be double-stranded or single-stranded. Similarly, ribonucleic acid generally it is single-stranded, which have function in the nucleus of the cell. DNA, it consists of nucleotides. The structure of a nucleotide is composed of a sugar molecule, which is deoxyribose, phosphate sugar, and different type of nitrogenous bases like adenine, guanine, cytosine, and thymine. A very important point in double-stranded DNA is that the two strands are held together by hydrogen bonds. This helps in storing and transmitting genetic information, whereas in single-stranded molecule, which is RNA, again it is consisting of nucleotides. The difference is that instead of thymine, here the nitrogenous base is uracil. These are very important in protein synthesis, RNA. So we have messenger RNA, which carries genetic information from DNA to ribosomes. We have the transfer RNA, which transfers amino acid to ribosomes during protein synthesis. Ribosomal RNA, which forms the structure of the ribosomes. Also important to know are some characteristics of nucleic acids such as denaturation, melting, and apart from that, even the dimensions of the different types of components, the dimensions and forms of ABZ forms of DNA, but that is beyond the scope of this video. Maybe we can make another video for the same. Next concept is metabolism. Metabolism consists of two components, which is catabolism and anabolism. Catabolism means breaking down complex molecule into simpler ones. Examples are cellular respiration and digestion. In this regard, you also have to study carbohydrate metabolism, which includes glycolysis, TCA cycle, electron transport chain, ATP synthesis, and also do know about the glycogen metabolism, vis-a-vis the synthesis and the degradation, what are the enzymes which are involved, and what is the hormonal control for the same. Anabolism is the building of complex molecules from simpler ones. Examples will be biosynthesis and photosynthesis. Here you would have to know about the nucleotide biosynthesis, which is purine and pyrimidine, both de novo and salvage pathway. Also the metabolic pathways, other ones, try to know the different type of components, the intermediates which are present in the pathway, the cofactors which is required, and along with that, very important, the regulation. We know that the enzymes are tightly regulated to maintain homeostasis, especially the regulatory enzymes. So here you would have to know about different kind of regulation like allosteric, feedback, covalent modification, and protein-protein interaction. This kind of regulation can occur at various levels, including gene expression, enzymatic activity, and hormonal control. Next concept to be covered is thermodynamics. So thermodynamics, what you have to know for exam are the important state functions like enthalpy, entropy, and Gibbs free energy. Enthalpy is the heat content of the reacting system, which is denoted by H. This is the symbol, and it is nothing but heat absorbed or evolved at constant pressure. This is the formula which you may be using for numericals, that the delta H or enthalpy change is equal to the internal energy change plus P delta V, where P represents pressure and V represents volume. What is entropy? Entropy is a measure of randomness or disorder of the system. If you have a crystalline solid, then it has perfect order in the molecule. It will be known to have low entropy. On the other hand, if you have some gaseous molecules, they will be known to have high entropy. This is denoted by the symbol S. If the change of entropy is positive, then the process will be favorable, which means that a system always moves to the increase in the entropy or increase in the disorderness. If delta S is negative, it means the process is non-spontaneous. If it is 0, it means the process is in equilibrium. Here it is important for you to know about a formula, that is delta S is equal to the final entropy and the initial entropy, which will be equal to Q, or the heat content of the system, divided by the temperature. Next is Gibbs free energy. Gibbs free energy is the indication of the chemical energy which is available from a reaction. It is not internal energy, it is not activation energy, but it is the energy which can be freely removed or added to a certain molecule. How we denote is this with the letter G. The formula for this is delta G is equal to delta H minus T delta S, where delta G is the change in free energy, delta H is change in enthalpy, T is temperature, and delta S is change in entropy. This helps in predicting spontaneity of a particular process. For example, if the change of free energy is negative, then the process is spontaneous. Always remember in biomolecules, if for any molecule the energy is less, then that process will be very much stable. Then that molecule will be very much stable. And whatever processes go towards lowering of energy, those processes are spontaneous processes. If delta G is positive, then the process will be non-spontaneous, and if it is 0, then the process will be in equilibrium. There are certain standard conditions like delta G of reaction. It depends on some conditions. Standard state has set reference conditions. For example, temperature will be 298 Kelvin, pressure will be 1 atmosphere, and concentration will be 1 molar. Here you have to know this particular equation. The change in free energy change, if you need to calculate in case there is some condition changes from the standard state. So you have to take the standard free energy plus whatever has changed, that is RT ln product concentration divided by reactant concentration. Remember that concentrations other than 1 molar will change the value of delta G. If you have to calculate it, then you need to apply this formula. A small tip and trick over here is that pay attention to the units, learn about the log rules, and also write down the formulas in one summary page. That way you will be able to apply them in whatever numerical question is asked. At equilibrium, delta G is 0. Thus, delta G0 is equal to minus RT K equilibrium. Remember, delta G0 and K equilibrium are inversely proportional to each other. Not only standard state, but there is something called as a biological standard state. The same formula will be applicable over here. It's just that we represent it with a dashed. What is biological standard? We also take the pH into consideration. But for that, the hydrogen and concentration cannot be 1 molar, but instead it is 10 to the power minus 7 molar. Next, let's move on to enzymology, a very important component of biochemistry. So, we will be looking at the enzyme structure first. Enzymes are typically composed of one or more protein chains which fold into complex 3D structures. Here, the active site of an enzyme is where the specific substrate binds and the catalysis will be taking place. Different enzymes may have distinct structural features like domains, motifs, cofactor, binding sites, etc. Coming to the Michaelis-Menten equation, here the different terms are important. So what you can do is you can write down the terms and what is their significances and also the formulas which are associated with them. Learn about the unit conversions as it will be really helpful for numerical problems. Here kcat is what is called as the turnover number. It is called as the rate constant of the rate limiting step or the slowest step in the enzymatic catalysis. This is a first order rate constant, which means that the number of kcat, the value of kcat depends on the change of one of the components in the enzymatic reaction. Also called as turnover number, which is the number of moles of substrate converted to product per time per enzyme active site. They have units of reciprocal time which can be either within minutes or within seconds. Then we have the Michaelis-Menten kinetics, which we take at low substrate concentration. The velocity or reaction velocity is equal to the K2 or the rate constant of the rate limiting step into the enzyme substrate complex concentration. But when the concentration of substrate is very high, then all the enzyme sites are saturated with substrate. At that particular time, enzyme is working at full capacity or velocity will be equal to the Vmax. All of the total enzymes at this point are tied up as enzyme substrate complex. That is, enzyme total will be equal to enzyme substrate. If we can modify this equation at high substrate concentration, then what we can write is Vmax is equal to kcat into ET. Remember, this is a very important formula. From this formula, you can calculate kcat or ET given if Vmax is given or any other term provided the other two are given. Then next is the Michaelis-Menten equation for one substrate catalyzed reactions. V0 is equal to Vmax into S divided by Km plus S. Over here, this Michaelis-Menten describes a rectangular hyperbola if you take a graph of it. Exception to Michaelis-Menten kinetics are the regulatory enzymes. Here, what is the Km? Km is called as the Michaelis constant. It is the apparent dissociation constant of the ES complex, and this Km is equivalent to the substrate concentration which we require to achieve half of Vmax. Remember, Km is inversely proportional to the substrate's affinity for the enzyme. Specificity constant. It defines the catalytic efficiency of the enzyme, or how fast and how efficiently an enzyme will be able to convert substrate into product. This is calculated by the formula Kcat by Km. Coming to the next topic, that is amino acids, again an important topic for CSIR-NET. Looking at the general structure, it consists of a central carbon alpha, which is surrounded by 4 constituents. One is the alpha amino group, alpha carboxyl group, hydrogen, and a side chain which is variable. These are small organic molecules. The key elements are carbon, hydrogen, oxygen, and nitrogen. All amino acids found in proteins, they are alpha amino acids because the amino and carboxyl group are attached with the alpha carbon. Next, coming to isoelectric point, or it is the pH at which the amino acid exists as zwitterion. As you can understand that the amino group and carboxylic group can undergo ionization, and that's where their charge will change. If the amino acid is neutral, that means the positive charge on the amino acid equals the negative charge, then it is said to be in the isoelectric point. It is the pH at which the amino acid exists as zwitterion. What is a zwitterion? Well, it is a dipolar ion of an amino acid with spatially separated positive and negative charges. It can act either as an acid or a base, which we call it as an amphoteric compound. If the pH is above the pH point of an amino acid, then the amino acid will be having protons dissociated from it further, because increasing pH means lessening of H plus ion. Then the amino acid will have net negative charge on it. If the pH is equal to pi, then the net charge of amino acid will be 0, and at pH below its pi point, the amino acid will have net positive charge. The pi calculation is done by taking the average of pK values that involve the zwitterion. Ionization and pH. The amino acids, they will undergo ionization at different pH. For example, the amino group which is protonated will undergo deprotonation at a higher pH, and the carboxyl group will also lose its proton and become deprotonated at an acidic pH. So at acidic pH, the carboxylic group is protonated and amino acid remains in cationic form because the amino group still carries a positive charge. At neutral pH, carboxyl group is deprotonated but amino group is protonated. Hence, the net charge on the amino acid will be 0, and at alkaline pH, both groups are deprotonated. Thus, the amino group will be neutral and the amino acid will be in anionic form. The R groups also similarly get protonated or deprotonated. The value varies from amino acid to amino acid. So, we have different pK points for different side chains of ionizable amino acids like tyrosine, cysteine, lysine, arginine, histidine, aspartic acid and glutamic acid. Here you will have to memorize the pK points of the side chains of this amino acid, because these concepts are going to help you to calculate the charge, the net charge on the amino acid or on the protein. The last topic is protein structure, where you have 4 levels of hierarchy of folding. The primary structure consists of the amino acid sequence along with the polypeptide chain. So as you can see, this is what is the linear polypeptide chain. Here amino acids are joined by peptide bonds between carboxyl group of one amino acid to amino group of the another amino acid. This kind of reaction is called as condensation or dehydration synthesis. The linear sequence is always written from N to C terminal, and each protein has their own set of amino acids which are assembled in particular order. Coming to the secondary structure, they have distinct characteristic repetitive local conformation. Why local? Because this is formed in a continuous segment of a polypeptide. Repetitive because this keeps on occurring in several segments of the polypeptide, and they will have some particular type of structural conformation. So we have the stabilizing interactions between this is hydrogen bond. They are stabilized by regularly repeating pattern of hydrogen bond formation between the backbone atoms, and the side chains, they don't have any interaction in this particular structure. Next is the type of secondary structures. So we have the alpha helix, beta sheet, random coil, and the loops, which are not longer than 12 residues. Alpha helix, it is a coiled coil, mostly in right-handed form. The side chains extend out of the helical structure, as you can see from here. The dimensions you have to know that one turn of the alpha helix has 3.6 residue and it is 5.4 Armstrong in length. Hydrogen bonding is from the CO of the first residue to the NH of the I plus fourth residue. Coming to beta sheets, they are extended almost linear segments called beta strand. Adjacent beta strands hydrogen bond to form a beta sheet. Here you have the sheet conformation consists of pair of strands lying side by side. The R groups extend above and below the plane of sheet, as the R groups have no regard in the secondary structure. Beta sheets can be of two types, parallel and anti-parallel. The parallel beta sheets, their strand direction is from N to C terminus, which is same, and anti-parallel beta sheet, the strand directions will be opposite. That means one strand runs in opposite direction to the other. Remember, anti-parallel beta sheet is more stable. The reason is because they have more well-aligned linear hydrogen bonds, and linear hydrogen bonds are the most stable structure. Random coils, no regular secondary structure for them. They can be 4 to 20 residues long and not characterized by any regular hydrogen bonding pattern. Most commonly, they are found at terminal ends of the polypeptide. Apart from this, you have also different types of beta terms, but that is beyond the scope for this video. Let me know in the comment section which topic which I discussed today you want me to elaborate in my next video. Thank you very much.